From Microbe TV, this is TWiM, This Week in Microbiology, a special episode recorded in November 2016. Hi everybody, I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Today's episode is coming to you from Hamilton, Montana, at the Rocky Mountain Laboratories of the National Institutes of Health. Rocky Mountain Lab is part of the National Institute of Infectious Diseases of the NIH. It's located in Hamilton, Montana, a small community located between the Bitterroot and Sapphire Mountains. The facility was constructed in 1928 and is well known for its research into vector-borne diseases, such as Rocky Mountain spotted fever, Q fever, and Lyme disease, 3 illnesses caused by microbes whose names pay tribute to former Rocky Mountain Lab scientists who discovered them. Today, research at Rocky Mountain Labs includes other infectious agents like chlamydia, Q fever, Lyme disease, plague, tularemia, salmonella, prion diseases, and a variety of viruses. Rocky Mountain Labs encompasses roughly 30 buildings on 36 acres of land and employs about 450 people. The Rocky Mountain Laboratory Integrated Research Facility is the first NIH facility of its kind to house BSL-2, BSL-3, and BSL-4 laboratory space in one building, along with administrative offices and conference rooms. Robert Heinzen, my guest today, is investigating the host-pathogen relationship of Coxiella Burnetti, the causative agent of a disabling flu-like illness called Q fever. Coxiella has historically been considered an obligate intracellular bacterium and is a Category B biothreat. I met up with Robert during a recent visit to Rocky Mountain Laboratory. Listen as we talk about the history of Q fever and his work on the causative microbe Coxiella Burnetti. My guest is a senior investigator in the Coxiella pathogenesis section in the Laboratory of Bacteriology here, Robert Heinzen. Welcome.
Well, glad to be here.
Is that the right way to say it, Heinzen?
Yep, just like German pronunciation.
Heineken.
Yeah.
Where are you from originally?
I'm from central Minnesota.
You didn't go to Carleton College, did you?
No. I went to a state university, St. Cloud State.
St. Cloud State, right.
And then moved on to Washington State for my PhD.
So you knew right out of college you wanted to get a PhD?
I wasn't quite sure what I wanted to do, and then after having some long discussions with my advisor, we decided that the graduate school was probably a good idea to at least investigate. And I didn't know if I was going to go through with the PhD, but I definitely knew I was at least going to get a master's. So
right, and this is in University of Washington?
No, Washington State.
Washington State. Is that in Pullman?
Pullman.
That was a PhD program, and what were you going to get your degree? Was it microbiology?
It's microbiology.
Okay.
Mm-hmm. So I worked on Q fever, Coxiella, right from the get-go.
I was going to ask you, what led you to Q fever, but you just found a lab that you were interested in.
Yes. Well, I knew I wanted to work on a human pathogen, so there were 2 or 3 labs at the university that actually did that, and I found this particular bug to be rather fascinating because of its obligate nature at the time, and the fact that it prefers to invade and replicate in macrophages, which is unusual. And beyond that, it lives inside a phagolysosome, which is probably the only pathogen out there that does that.
So things like Legionella go into intracellular vesicles, but they're not lysosomes.
Well, most vacuolar pathogens have sophisticated ways of preventing lysosome fusion.
Right.
Because generally that's a toxic event. But Coxiella just goes through the default pathway and lives in this environment where it's acidic and the hydrolases are active, and it's an environment that if we introduce E. coli into this vacuole, it's basically destroyed in about 15 minutes.
Yeah. Most viruses, if they come in by the endocytic pathway, they escape before lysosomal fusion. But there are some that stay in, like reoviruses, and that's so the outer shell can be digested away, and then they penetrate through the wall after.
Yeah, that's a delicate dance, I bet.
It is.
Because you don't want to stick around too long.
Nature, evolution is very good at choreography.
Yeah. But we're trying to figure out how it can actually do this, how it can resist those digestive factors. And we know it has an unusual cell wall. And I have a fellow who is trying to understand the biochemistry of that cell wall and how it changes during the infectious cycle. And that's a difficult project that involves some pretty sophisticated mass spec procedures and identifying different crosslinks and so on. But that must be, obviously the resistance is due in large part to the outer envelope or the cell wall of the organism. And so if we understand that, then we can understand how it can live in this environment.
Now, when you started doing your PhD on this, so the organism we haven't even mentioned, it's Coxiella Burnetti. Mm-hmm. So I guess someone named Burnetti discovered it.
Well, it's actually Burnett, who, if you may not know this, is a Nobel Prize winner.
Macfarlane Burnet?
Exactly.
From Australia.
Exactly.
Okay.
He won the Nobel Prize for, I believe, his work on immune tolerance.
Okay.
But he, at the time, was, when Coxiella when Q fever was first described in Australia in the late 30s, he was brought in to try to understand what the infectious agent was. Because he was a rather established virologist at the time. And he was able to take some patient samples and infect guinea pigs, mice, and do some staining and see what he called rickettsia-like organisms inside small vacuoles. And at the same time, at the Rocky Mountain Labs, they were studying a pathogen called the 9-mile agent. Mm-hmm. And a fellow named Harold Cox, he's the other part of Coxiella Burnetti, he did some very nice work with that agent. And it actually caught the attention of a fellow by the name of Rolla Dyer. I don't know if that rings a bell.
It does, yeah.
The former head of the National Institutes of Health. And at the time, he was director of some institute at the NIH. And he came out here to visit Harold and talk to him about this new agent. And he actually became infected with the 9-mile agent.
Yeah.
And he recognized that his symptoms were similar to what they were describing for Q fever in Australia. So through the exchange of reagents, they established that this 9-mile agent that came from a tick in Montana and the Q fever agent in Australia were the same pathogen.
Right.
So it was actually a laboratory-acquired infection that was instrumental in establishing the etiology of Q fever, of none other than the director of the National Institutes of Health.
That's great.
Can you imagine that happening now?
No, they would throw you in jail for not having the right containment or whatever.
That would be the end of your career.
Yeah.
So that's where Coxiella Burnetti comes from.
So where did Q fever come from in that name?
Q fever is the original physician, the physician that first described Q fever as a clinical entity, Derek. referred to it as Q fever, and Q was short for query, because he didn't have any idea what was causing this disease.
Right.
He just called it Q, which was short for query.
So what is the disease like?
It's a severe flu-like illness, generally, the acute disease where you have a high fever, a very severe headache, and this will last for a week or more, but it generally resolves even in the absence of antibiotic therapy, and in rare cases, in association with some predispositions like heart valve issues, it can cause endocarditis. It can go chronic and cause some more severe problems that are more difficult to treat.
So there are no respiratory symptoms?
Well, yes, pneumonia. Pneumonia is probably occurs 30 to 40% of the time with acute disease.
Okay.
Yeah.
And fatality rate is about
low, less than 1%, untreated.
All right. So is it spread by ticks mainly, or respiratory?
Well, the tick is interesting because the original isolate here was derived from a tick. Right. So they were actually looking for ticks that were carrying Rickettsia rickettsii that cause Rocky Mountain spotted fever. And they would flag ticks, collect ticks, and bring them back to the lab, and they'd put them on guinea pigs and wait for the guinea pigs to get sick. So when one did, they would draw blood and reinfect and try to get the agent out of the spleen. Pretty classical stuff. So in one of these tick collection surveys, a guinea pig developed a febrile disease, but it wasn't Rocky Mountain spotted fever. And they know that because guinea pigs infected with Rickettsia rickettsii have this swelling of the scrotum that's very characteristic. And this guinea pig did not have that, but it still had a very high fever. So that's how they got the original isolate. It was from a tick, but I can't think of a single well-documented case of human tick-transmitted Q fever. So in terms of disease in humans, it's really not a factor.
It's respiratory transmission, right?
It's an environmental pathogen. Yeah. So the primary reservoirs are really domestic livestock, sheep and goats primarily, and dairy cows in this country. There are millions and millions of dairy cows infected with C. burnetti. And the CDC has looked at this recently, and they just randomly examine, bottled milk samples from all over the country for the presence of C. burnetti DNA. Mm-hmm. And something like 95% of these have evidence. So dairy cows are chronically infected with this.
Are they sick though? They're not sick.
They're not. Not in any measurable way. And so we pasteurize. And the reason the parameters for pasteurization are based on killing C. burnetti. Originally, they were based on killing Mycobacterium bovis.
Like the Pasteur original.
Right.
Yeah.
And it was recognized in the 50s that this was not stringent enough to kill C. burnetti, so they raised the temperature. So now you went from a lower temperature, longer time, to a higher temperature, shorter time that would kill C. burnetti. But what's interesting to me is the C. burnetti strains that were in dairy cows in the 40s and 50s seem to not be the strains that are in dairy cows now. There's been an ecological shift of some kind, and it may be that these strains in dairy cows are weakly pathogenic for humans, because you just don't hear about outbreaks associated with dairy operations the way you used to in the 40s and 50s.
So there used to be outbreaks on dairy.
In California especially, there were large outbreaks associated with the dairy industry. But they're not. If they're happening, they're not being reported, or they're not large enough to be noticed, or the strain just is not. It's pathoadapted to be cause chronic infections in dairy cows and not be highly pathogenic for humans. And we know there's strain variation among Coxiella, where we know we have strains that are highly virulent, moderately virulent, and show low variance, at least in animal models. So it may be that the strain in dairy cows now is just a low virulence pathotype. I just think it has to be because there are so many dairy cows infected with this, and they shed so much of this material into the environment that if it was truly infectious, you would know it.
Have people done serological surveys of dairy farm workers?
Yeah, they've done. I don't know if they specifically targeted that population, but they certainly look at veterinarians and people associated with livestock operations, and their titers can be as high as 30-40% serial positivity.
And the general population is much lower.
The general population overall is about 3%. But it really depends on where you're at, where in the country you're at. And, you get into some rural areas, it's going to be higher. I've always had a titer.
You worked with it your whole life, right?
Well, I had a titer before I worked with it, because in graduate school they would draw blood and do the serology. And they did that before I ever handled the organism. So I came out of central Minnesota with relatives who, where we drank unpasteurized milk and played with the goats and that kind of thing. And I'm pretty sure I just picked it up as a kid.
So is it limited to the U.S. or is it a global distribution?
No, it's everywhere on the planet except New Zealand. And they very much want to keep it out of New Zealand because they have a very large sheep industry there.
Right. They must have cows also, right?
Yeah. And then you have Australia that still has actually a fairly major problem with Q fever, to the extent that they still vaccinate thousands of people a year there.
Really? So there's a vaccine at least in Australia, but not here in the U.S.?
It's not licensed here, and it's unfortunate because I think there would be at-risk populations that might want the vaccine, especially people working in a lab like we do. And it's a very, very efficacious vaccine. In fact, I would challenge you to find a vaccine that's more protective.
Yeah.
It's near 100%.
Yeah. You said now here in the U.S. the virulence of the strains seem to have decreased. Is that also the case in Australia? It sounds like they continue to.
Well, I was speaking specifically about maybe some of the strains that are in dairy cows, but I'm sure there's still virulent strains circulating in the U.S. And in Australia, I imagine that the strains there are also. I don't know of any information that says they're less virulent.
So there are no outbreaks in the U.S.
There are small outbreaks.
Small ones.
15, 20 people
okay.
at a time, and they're generally associated with small dairy goat operations or moving animals from one place to another and introducing an infected animal into some population naive animals, and then you have naive people working with them. That's generally how it happens. But in the Netherlands, I don't know if you're aware of this outbreak that occurred from about 2007, I think, to about 2011. They had over 4,000 cases.
Yeah. Wow.
And that was attributed to a very large dairy goat operations that the industry kind of exploded, and a lot of these farms were close to some pretty major metropolitan areas. And just through wind dispersion and people coming and going from these farms, they had a very large outbreak, which was very, very expensive to deal with. And I think they estimate it cost over $1 billion to.
When was this?
From about 2007 to about 2011 or 12. So they had to cull, I believe, over 60,000 pregnant dairy goats, because the largest numbers of C. burnetti that are put into the environment are through birth products. So there's aborted that can cause abortions in goats and sheep, and these infected placentas are 10 to the 9th organisms per gram. And you have a pathogen that has an infectious dose of less than 10. You can do the math and figure out how much one placenta could infect thousands and thousands of people.
Is this the case also with cows, pregnant cows? They have a lot of causes abortions in them as well.
Well, they don't seem to cause abortions in dairy cows like they do in goats and sheep. It obviously is not an economic burden, because there doesn't seem to be any effort to eliminate this pathogen. In the Netherlands, I believe they started vaccinating their dairy goats, but there's nothing like that going on with dairy cows that I'm aware of.
Yeah. Now, aside from cows and goats and sheep, is there a reservoir in nature that carries?
Well, when this sort of work hasn't been done for a while, but when it was back in the 50s, you can find evidence of infection in just about every animal you look at.
Okay.
From fish to birds.
Wow. To
rodents to various arthropods and so on.
Okay.
So it's a very successful pathogen. Of course, it doesn't. Pathogenesis is an accident, like you said in your seminar yesterday. It's not there to cause disease; it's there to replicate, and it can replicate in just about anything.
Amazing.
Yeah. So, I mean, the zoonotic cycle isn't all that well defined. I mean, there must be some cycle between ticks and small mammals that keep it going in nature, and then maybe that's how some of these naive livestock herds can get infected that way, and then they spread it. Then the spread can become aerosolized.
You don't go out and collect ticks and look for it, right?
No, not anymore.
People used to do that, though. But you said they never found it in a tick other than that one time, right?
No, you find Coxiella in ticks. It's not that hard to find. It's just, it doesn't seem to be that transmissible, at least to humans. And the other confounding thing about Coxiella and ticks is that ticks are loaded with Coxiella-like endosymbionts. So you need to be careful about what it is you're detecting.
I see.
So you need to design your probes so you're not detecting the endosymbionts, but the actual pathogen.
But Coxiella is not an endosymbiont.
No.
Right. Okay. So it's called a Category B biothreat.
Right.
Well, it sounds to me that there's less than 1%.
Well, yeah. I mean, we could spend 3 days talking about this. I mean, unfortunately, it was experimented with by the Army and the
tried to weaponize
for quite a while. Well, yes, and not clear to me what weaponization actually means. If it means growing up a lot of infected yolk sacs and diluting it with PBS and putting it in something that sprays it, I guess they did that. But it was part of this covert biowarfare program or biodefense program that was called Operation Whitecoat. Are you familiar with this?
I've heard of it, yeah.
Right.
Yeah.
So this went from the, I believe, the late 40s into the late 40s, early 50s into the early 70s, and it involved human volunteers that would volunteer for experimentation with these putative biowarfare agents, Coxiella, Francisella, Bacillus, Yersinia. And these were largely Seventh-day Adventists who were very patriotic and wanted to volunteer for military duty, but would not take up arms and commit, violence.
Yeah.
But they would volunteer to be have their face put on a mask that would aerosol in pathogens. And Coxiella was one of those pathogens. That's why we know the human infectious dose, which you don't know for too many pathogens.
Yeah.
This was all done at Dietrich with something called the 8-ball. Have you heard of the 8-ball?
I have not. No, that I have not. What's that?
Well, the 8-ball at the time, anyways, was the largest aerosolic generator ever built. And it was built to aerosol various pathogens, and it was designed so that you could challenge
I see.
human by the aerosol route. Yeah. So it's this massive aluminum sphere, and they had respirators or masks that these volunteers could put their face on, and then they would just inhale these organisms. They knew approximately what the dose that they had acquired, and they would do that to test vaccines. They do that to test treatments. They do that to understand the infectious dose. So Coxiella was kind of a big part of this, and I really wonder if it was just used as a surrogate, because it's not that lethal of a pathogen, but it's a very good pathogen to test aerosol dispersal of something that might be actually worse. And they actually did this in the desert in Utah. They had a test where they put volunteers and guinea pigs and monkeys about more than a half mile away from an aerosol generator, and then just turned it on, and it drifted downwind, and they were breathing normally. And after this test had finished, they all went back to the airfield, and the story goes they took their clothes off, burnt their clothes, and then they flew them back to Fort Detrick and waited to see if any of these individuals became sick. And most of them did.
Some of them probably died, right?
No.
None of them died?
No one actually in all of Operation Whitecoat, there were no deaths.
Okay. And it was called Whitecoat as opposed to them, because they didn't go in the army, right? I guess.
There is a story behind why they were called Whitecoats, but I don't recall what it is.
So, what does category B mean? So, I know there's A, B, and C.
Well,
right?
Yeah, I think that's an NIH classification or a CDC's classification.
Right.
So you have select agents.
Right.
And then within the select agent grouping, you have, there's A, B, and C pathogens, category pathogens. And it has to do with a number of factors, and how easily are they transmitted? Are they contagious? What's the severity of disease? So you have your A's that are pestis, Ebola.
Those are the worst, right?
Anthracis, tularemia. There really aren't that many. And then you have the B team. Coxiella's on the B team with, you're going to pull these out of my head here.
So yours is in there, Coxiella.
It's a B, yeah. And there's actually C's, and I believe some of the enteric pathogens, but those are not, there's no category C that is actually a select agent.
Okay.
There are B's and A's. All A's are select agents, I think, and most B's are.
What does it mean to be a select agent, that it's a certain category that they consider
The select
dangerous?
agents are regulated
Okay.
extensively by the CDC. So there are a number of rules and regulations that have to be followed to actually have possession of this and this pathogen, and the facilities requirements to work with it. There are a lot of legal issues related to working with these pathogens that you have to follow the rules or it's not a good day.
So category A's have to be worked on in a BSL-4?
Not all of them.
But Ebola certainly does. Right?
Yeah, so there are category A's that are level 3s. And those are primarily bacterial, I believe. So, Bacillus anthracis is a 3. Tularemia is a 3. Pestis is a 3. I don't know what viruses are in there, but there are some.
If Ebola is in there, that would be a 4.
Yeah.
Yeah.
Right. There aren't that many select agent 4s.
I think Crimean Congo hemorrhagic fever virus, Lassa.
I think Lassa is actually
It's a 3. No, it's okay. But I think also Marburg virus is a 4 as well. Yeah. And Coxiella is what?
It's a level 3 pathogen.
3. Okay.
Yeah.
So in this building, you have to go to the.
We go to a different building that has engineering to allow you to work safely, and a lot of it has to do with restricted access, has to do with airflow, it has to do with anti-rooms where you can put on PPE and take off PPE, pass through autoclaves, that kind of thing. And people obviously studied these pathogens for decades without any of this.
Yeah.
But there actually were large outbreaks, laboratory-acquired outbreaks of Q fever in the 40s and 50s. And at NIH, it was quite a problem. They had outbreaks with dozens of people on the main campus.
Okay, so that's why it's a 3.
Well, it's a 3 because,
it's easy because it sounds like it's easy to infect yourself, even if
it's
you're being
very,
careful.
very easy to infect yourself. So, we wear respirators. But again, this is all, in graduate school we didn't. We put a lab coat on.
So when did it turn into a level 3? Roughly?
Well, it's been level 3 since those levels were established, and I don't know when those levels were established. The select agent program, I believe, started in 2002, and then it became a select agent. Right.
So when you were a PhD student, you worked on Coxiella, and then you did a postdoc also?
I did a postdoc here.
On Coxiella?
Well, actually on mostly on rickettsia, mostly on spotted fever.
With whom?
With Ted Hackstead. I don't know if you know Ted.
I don't know.
Yeah.
So another intracellular bacteria.
And I dabbled also in some chlamydia, and I did do a little Coxiella work here as well. So I ended up having experience with 3 different obligates that I felt gave me some flexibility when I left to decide what to try to build a program around.
But you didn't leave, right?
I did.
Oh, you did? And then you came. Where did you go?
I went to the University of Wyoming for 7 years as a tenure track assistant professor, and I got tenure.
What did you work on?
Worked on primarily Rickettsia. And towards the end, a little bit of Coxiella, because we got a grant to do it. And then the biodefense initiative that was spawned by, in large part, the anthrax release, 9/11, that sort of thing. And as you know, they put a lot of money into biodefense research back in, or 2003, 4 or 5 years there was pretty good. And part of that initiative was to, at least within intramural NIH, was to get people back here working on some of these pathogens. And so nobody had worked on Coxiella here for 20 years. And we had a strain collection that was just out of sight.
Here? Yeah,
which is largely gone now, because I'll tell you, because of the select agent program. It's gone because there's inventory requirements, and I was not prepared to count every vial in 3 Revcos.
So in other words, to be compliant, you had to have an inventory of every vial. You had to know what was in it and where it came from?
Well, these inventory requirements are interesting. You need to know the number of vials you have.
Okay.
But you don't need to know how much material is in the vial. Mm-hmm. And you don't need to know how infectious the material is in the vial. You just need to know how many vials you have. So we count vials. When we count them, and we count them, and we count them. And then we walk out and we go, well, there's no germs missing. And we all know how silly that is. But that's the rules. Those are the inventory requirements. And so I've had to downsize our inventory to something that's manageable. But if we didn't have these regulations, I would have kept everything. Because I think, the minute you throw something out, you'd think, I wish I had that. A lot of it was passage history variants,
right.
In past different times. But, it's just, when you have to count everything, it would take hours. When we started, it took hours to do this.
I'm sure.
We're doing this 4 times a year. So now we're down to something that, we can get through the inventory in a half hour.
Oh, so you have to do inventories 4 times a year, not just once at the onset.
No.
Wow,
no.
I see. I thought that's different.
Well, those are NIH rules.
Yeah.
I don't know what the official selected agent rules say. You have to know what you have.
Yeah.
I don't think they tell you how often, or even if you have to count them. So we do complete inventories twice a year and partial inventories twice a year.
So,
but we managed to make it simpler by if you tape boxes with special tape,
right,
with numbers, with security tape, you can count the box, and you don't have to count all the tubes in the box.
Okay. So when you came back here because of the biodefense initiative, right?
Well, then, they had an opening for somebody to work on Coxiella.
So you came back, and that's when you had to pair the collection. Right.
And, we actually had it pretty good in Wyoming. I had some pretty good funding, and things were working fairly well there. But one thing I realized once the select agent program started was that there was no way that this level 3 lab that I'd cobbled together down there was going to pass inspection. Mm-hmm. So they would have shut me down. And you can see the infrastructure we have here. We have multi-million-dollar facilities to work on these pathogens, plus all the security that you now need.
Yep.
And that just wasn't going to happen at little University of Wyoming.
No, that's It's a government thing. That universities typically can't do that. Yeah, I totally understand, having been at a university all my career.
Well, I think universities might be reluctant to even dabble in some of this because there's liability issues. There's, administrators want to know if they're going to get their money back on something.
Sure.
And if the select agent program has all this overhead associated with it and they're not getting a return, then they won't want to deal with it.
So one thing I wanted to talk a little bit about is this is an obligate intracellular bacterium, and I know for years it could not be grown in culture medium, and you took care of that. So can you tell us that story?
Well, again, it had to do with coming back here, and at about that time, the genome was deciphered in 2003. So we got to look at the genome, and when you looked at it, you go, well, there's nothing in this genome that indicates why this would have an obligate intracellular nature. It has all the path. It has some defects. It's oxytrophic for some amino acids, but it has all the enzymes you need for intermediary metabolism, and so that along with some new techniques we had now, such as transcriptional microarrays and metabolomics, we decided to take a long-term look at whether or not we could develop a media that essentially was close enough to that phagolysosomal media, or milieu, that would satisfy this pathogen and get it to grow outside of a host cell. So I got a new postdoc who was originally from Norway, Anders Omsland, and I sat him down and convinced him that this was a good project. And I convinced him that even if we can't get it to grow, the metabolic insight that we gain will be publishable. What is it used to become metabolically active in a test tube? So it was a matter of just starting from the very beginning. And for example, the first thing Anders did was to find a good buffer. And his measure of metabolic fitness was the ability of Coxiella to incorporate 35S-methionine into protein. Can it make protein? And so when he did the assays, he would throw a little glutamate in there because that would allow it to make some protein. So he would look at protein synthesis, and it would add things and see if it got better or it got worse. So it was add a little this, add a little that. And we came up with a really good buffer that was, allowed for like 100-fold greater protein synthesis than some of the early buffers. But when we looked for replication, it was obviously not replicating. So, when you get to a point like that, you have to ask, what else could it be? And what haven't we tested? So to make a long story short, it turns out that we hadn't really tested oxygen tension. We just assumed that this was an obligate aerobe. Because it grows in a mammalian cell.
Right.
And we're aerobes. But then you have to think about tissues and what's the effective oxygen concentration in a macrophage in a lung.
Right.
It varies quite a bit. So he's titrated out metabolic fitness under different oxygen conditions. And when we got down below about 5% oxygen, it just took right off and started replicating.
How did you reduce it in some kind of chamber of?
We have these tri-gas incubators where we replace the oxygen with nitrogen, and they can be dialed down to less than 1% oxygen.
Wow!
So it was the oxygen.
So how long did the project take from start to finish?
I'd say to see actual replication, it was about 3 years.
Wow.
But now we've gone through 3 iterations of the media. The first media was good. The third media is much better. Now it's completely defined.
But it still has to be grown under low oxygen,
Yes,
right?
yes. And the media is really just 20 amino acids. And another trick was the addition of beta-methyl cyclodextrin and the addition of a lot of cysteine. We honestly don't know what benefit Coxiella is getting from these components, but these are things that have been added to media of other fastidious bacteria. And cyclodextrin is known to sequester toxic molecules, especially lipid-type molecules. So it might be a way to eliminate something that the pathogen doesn't like. And the cysteine, it may be that it's a cysteine oxytroph, and the buffer is actually the pH of a phagolysosome. It's pH of about 5. So at that pH, cysteine converts to cysteine, a dicysteine, and I don't think the bacterium can transport that and utilize that. So the effective concentration of cysteine is low at low pH. The available,
yeah, because most of it's dye. Interesting. So you can grow it in broth. Can you grow it on agar plates?
Yep. And very tiny colonies.
It's colonies. So you can do genetics then, right?
Yes. So that was the most immediate benefit of azenic growth. Was another very talented postdoc, Paul Baer, just essentially turned it into E. coli in terms of genetics.
Yeah.
And we have just about everything you have for E. coli now. We have shuttle vectors, we have transposons, we have reporter systems, you name it. So
that was the Norwegian postdoc happy?
He was happy. But I have to tell you, I didn't believe him for about 6 months. I just thought it was a contaminant.
Interesting.
And he'd show me this media, and it was cloudy.
Yeah.
I go, well, take it down to EM. Let's take a look at it. And I had him send the media to other labs that grow on Coxiella. And a trick you could do without these incubators, I don't know if you've ever heard of a anaerobe jar. Well, you put cultures where you want to grow anaerobes or microairfields into this jar, and then you have a packet in there that creates a reaction and eliminates oxygen.
Yeah.
So you can get it down to about 7% or 6%. Mm-hmm. So we sent these jars in the media to other labs that had Coxiella to see if you can see if something will grow in this.
Yeah, it did.
I I really had to know. And it was definitely growth. It was, it was 3 logs of growth. So, and what we got out of it was infectious. So. And
what year was this accomplished?
I think it was 2009.
I see. Not too long ago. By the way, are there viruses of bacteriophages that infect chlamydia?
Not that we can see.
Okay. So, but you can use plasmids and shuttle vectors to manipulate.
We do everything with electroporation. We haven't found the need to get more sophisticated than that because we can do targeted gene inactivation. We can do what we need to do. We have a Crelock system now, so we can take out large parts of the genome.
Right.
With the defined media, we now have non-antibiotic selectable markers. So we take advantage of some of the oxytrophies that the pathogen has. Right. So it can't make glycine, for example, but it has a number of the enzymes in that pathway except the last enzyme. So we can complement with a plasmid that encodes that last enzyme. Mm-hmm. in the presence of media lacking lysine.
Right.
And I think we have another one for arginine as well. Another issue with the select agent program is they approve what antibiotic resistance markers you can put into these pathogens, and you're limited. I mean,
Yeah, that's right. Yeah.
We're lucky that we have the 3 that we have. So coming up with something that's not antibiotic based is valuable for us.
Now, you mentioned at the beginning that the capsule is important for withstanding the lysosomal. Can you knock the genes out that are responsible for the synthesis and then show that it can no longer survive in a lysosome?
Well, it doesn't necessarily have a capsule that we're aware of.
Outer membrane?
It has an unusual cell wall structure.
It's a gram?
The gram negative.
Negative, okay.
So the peptidoglycan we're finding has some unusual properties. And unfortunately, the synthesis of that molecule is actually quite complex, so it's be hard to know what. And we have some mutants in cell wall biosynthesis, and some of them do have defects for growth, but they're not real strong.
Okay. So what are some of the key questions you have now on Coxiella?
Well, to me, the most interesting questions are how does it survive in the lysosome? Mm-hmm. And again, it comes down to resistant. This thing is very resistant intracellularly and extracellularly. And there's a developmental cycle that the organism goes through where you have the replicative morphological form looks very much like, a typical E. coli as far as ultrastructure. But once they get in the stationary phase, they really condense. The cell wall gets very thick, and the chromatin condenses. And that's not something you see very often in bacteria is a nucleoid that is extremely condensed. So we're trying to understand the significance of this developmental cycle to resistance in the lysosome and resistance in the environment, because it can remain viable in the environment for perhaps decades. So it's almost like a spore, but not quite. So with genetics, we're trying to establish what factors are involved in this. The other thing we've become interested in recently is the genetic basis of strain variation in terms of pathogenicity. Mm-hmm. So, like I said, in our collection, we have strains that have very different ranges in virulence. And it turns out that the strain that is least virulent has the largest genome. And phylogenetically, it looks like this is the more primitive strain. And as these organisms got involved with other hosts, virulence was associated with gene loss as opposed to gene gain. And so we would like to take this primitive strain and understand what is it about the unique genes in this strain that presumably make it less virulent.
Right.
So more anti-virulence than virulence. So now we can, and unfortunately the novel genes largely occur in blocks. So we would like to be able to take out these blocks and see which ones are responsible for this attenuation.
Right.
That's another project. And then the organism has one virulence factor we know is absolutely critical is it has what's called a type 4 secretion system. And you're probably aware of the different secretion systems in bacteria. There's 1, 2, 3, 4, 5, 6. I don't know how many there are now. It might be 8. But these are what are called molecular syringes, where they have the ability to inject proteins with effector functions directly through a eukaryotic membrane into the cytosol. And these effector proteins have an amazing diversity of functions. And in Coxiella, we've identified about 140 proteins that are secreted. Now, understanding what they do is a monumental task. And I believe that a lot of them probably do nothing, that they're redundant, or they're in the process of being removed from the genome. So what we're focusing on now are the effector proteins where the genes are full length in every strain that's been sequenced, so the conserved core. And we know of the function of a few of them, but we'd like to know more about what they do, because we know if we take out the secretion system, you have a completely avirulent organism. It can't grow in cells at all.
If you take the secretion, if you take the effectors out.
Well, if you take
the
take out the system that secretes the effectors, it's basically a null mutant. And we have mutants in some of the individual effectors, and those have replication defects, but they really don't approach the defect of the secretion system itself.
Yeah. Now, you said that loss of genome is associated with increase in virulence.
Well, that's a hypothesis.
Right. So are any of these effectors part of the genes that are lost?
So what we're doing now is we are screening the novel gene content of this. It's actually the strain is called Dugway, and it was isolated in Dugway prior to the release of Coxiella in this bio test.
Yeah.
They did a survey of the area to see what was already there.
Right.
So we have an isolate from a rat called Dugway. And Dugway has the largest genome, and when you put it into a guinea pig, it does almost nothing.
Right.
So we are trying to find out we've cloned all the novel genes that make sense to clone, and we're screening them for being a secreted effector protein.
Okay.
So we're going to find the novel effector proteins of this pathogen. And then hopefully it's reasonable to go back to the strain and take out one or more of these and see if increases somewhat in virulence.
Right. So your animal model is the guinea pig then?
The guinea pig's a pretty good model.
Do you put it intranasally?
Well, we're not going to do that just yet. We'll just go IP and see what happens.
But that's not a respiratory infection when you do that, right?
No, but that historically is how it's been done.
Okay.
We could look at more of a respiratory route, I guess. We at one time were going to utilize a whole animal exposure chamber.
Right.
And I got cold feet about using that for a number of reasons. It seemed like there had been some problems with it at other institutes. And
so when you put the organism's IP into a guinea pig, they get into macrophages?
Yeah. You get a very large splenomegaly out of it. So they get into the peritoneal macrophages, and those end up in the spleen, and they get a very nice fever response that is really recapitulates the fever response you see in humans. So mice don't really get sick from C. burnetti. They'll get a big spleen, but you won't see much else.
Do the macrophages ever get into the central nervous system?
Trying to think of neurological issues associated with. I mean, there are rare instances of just about everything. Bone infections.
Okay.
I'm sure there's some rare neurological complications.
Yeah.
I just don't know what they are.
People, if there are neurological complications, they're rare, probably. Right?
They're extremely rare. Yeah.
Okay. Is there anything else that we didn't cover that we should talk about?
Well, we covered a lot.
You did. It's a great overview.
I don't know. I think I like the field has really expanded.
That's what I wanted to ask. Yeah. So
I think I want to take some credit for that.
Sure.
Because it expanded because now people see this as an amenable model system.
Mm-hmm.
In the past, it was just a pain in the ass organism. You can't grow it. You can't do genetics. Why would you want to mess with it?
Right. No, you
and
should have credit.
And now you can.
Yeah.
And then now you can study a pathogen at a molecular level that lives in a macrophage inside a lysosome. So from a host-pathogen interaction standpoint, it's a fascinating model. And a lot of people have picked up on this. And we have sent plasmids and strains all over the world to labs that are, some that are old labs that are getting reestablished, some that are new labs. But, I think the number of labs working on Coxiella have expanded fivefold since say 2009, 2010.
Great.
And I think it largely has to do with, being able to do real science with it.
Sure. So do you have a Coxiella meeting?
We don't have a Coxiella meeting. We have a Rickettsia meeting that
you go to. And
Coxiella really doesn't even belong in this meeting because it was historically referred to as a Rickettsia, but it's not. It's a alpha proteobacteria. It's a gamma proteobacteria, and the Rickettsia are alphas. So genetically, they're not related. This is just because it was studied like Rickettsia, and it was the original name for Coxiella Burnetti was Rickettsia dioporica. That's what they called it here for a while. It was because it could pass through filters that normally would trap bacteria.
0.2 microns?
Well, there's these old Birkeland filters.
Yeah.
So they thought, again, initially didn't know if it was actually a bacterium or a virus or a Rickettsia or what it was. Here we are. Now we know it's just another bacterium that has its own special niche and
great story.
Very successful.
Great story. Coxiella Burnetti. My guest today has been Robert Heinzen from the Rocky Mountain Laboratories in Hamilton, Montana. Thank you very much for talking with me.
Yeah, it's great.
You can find TWiM at iTunes and at microbe.tv/TWiM. And of course, if you listen to podcasts on your smartphone or tablet, there are plenty of apps that you can use to listen to podcasts. You just subscribe to TWiM and you'll get it for free every time a new episode is released. Consider becoming a supporter of TWiM and the whole Microbe TV family of science shows. Go to microbe.tv/contribute. There are a number of ways you can help us out with our expenses, travel costs, and developing new shows. And we always love getting your questions and comments. Please send them to TWiM at microbe.tv. My thanks to Robert Heinzen for taking the time to speak with me during my visit to Rocky Mountain Laboratory, and also to Marshall Bloom, a virologist there who invited me to visit last fall. You might be interested in an episode of This Week in Microbiology that we recorded there with local high school. You might be interested in an episode of This Week in Microbiology that we recorded there with local high school science teachers and some of their former students who have gone on to careers in science, some at Rocky Mountain Laboratories. That episode, called Small Town Big Science, can be found at microbe.tv/TWiM/TWiM-140. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.